Buprenorphine, samidorphan, developmental code name ALKS 5461, is a combination drug formulation of buprenorphine and samidorphan acting as a kappa opioid receptor core antagonist which is under development by Alkermes as an adjunct to antidepressant therapy in treatment-resistant depression (TRD). Top-level data from two Phase 3 trials was released in 2016. ALKS 5461 failed to meet its primary efficacy endpoints. On the basis of a third Phase 3 study that did meet its primary efficacy endpoints, Alkermes initiated a rolling new drug application with the FDA. In late January 2018, Alkermes submitted a new drug application for ALKS 5461 to the FDA for the adjunctive treatment of major depressive disorder. A PDUFA date of 31 January 2019 has been set by the FDA for approval of ALKS 5461, i.e., the FDA will review and approve or reject ALKS 5461 by this deadline. Pharmacology. Pharmacodynamics. ALKS 5461 is a 1:1 ratio combination of 1-buprenorphine, a weak partial agonist of the mu-opioid receptor, MOR, antagonist, very weak partial agonist of the kappa-opioid receptor, KOR, and, to a lesser extent, antagonist of the delta-opioid receptor, DOR, and weak partial agonist of the nociceptin receptor, NOP, and 2, samidorphan, a preferential antagonist of the MOR, but also, to a slightly lesser extent, weak partial agonist of the KOR and DOR. The combination of these two drugs putatively results in what is functionally a blockade of KORs with negligible activation of MORs. Kappa-opioid receptor antagonism. It has been known since the 1980s that buprenorphine binds to at high affinity and antagonizes the KOR, through activation of the KOR, dynorphins, opioid peptides that are the endogenous ligands of the KOR and that can, in many regards, be figuratively thought of as functional inverses of the morphine-like, euphoric and stress-inhibiting endorphins, induce dysphoria and stress-like responses in both animals and humans, as well as psychotomimetic effects in humans, and are thought to be essential for the mediation of the dysphoric aspects of stress. In addition, dynorphins are believed to be critically involved in producing the changes in neuroplasticity evoked by chronic stress that lead to the development of depressive and anxiety disorders, increased drug-seeking behavior, and dysregulation of the hypothalamic pituitary-adrenal (HPA) axis. In support of this, in knockout mice lacking the genes encoding the KOR and/or prodynorphin, the endogenous precursor of the dynorphins, many of the usual effects of exposure to chronic stress are completely absent, such as increased immobility in the forced swimming test, a widely employed assay of depressive-like behavior, and increased conditioned place preference for cocaine, a measure of the rewarding properties and addictive susceptibility to cocaine. Accordingly, KOR antagonists show robust efficacy in animal models of depression, anxiety, anhedonia, drug addiction, and other stress-related behavioral and physiological abnormalities. A mouse study found that knockout of the MOR or DOR or selective pharmacological ablation of the NOP did not affect the antidepressant-like effects of buprenorphine, whereas knockout of the KOR abolished the antidepressant-like effects of the drug, supporting the notion that the antidepressant-like effects of buprenorphine are indeed mediated by modulation of the KOR by the drug, and not of the MOR, DOR, or NOP. However, a subsequent study found that the MOR may play an important role in the antidepressant-like effects of buprenorphine in animals. Buprenorphine is not a silent antagonist of the KOR but rather a weak partial agonist. In vitro, it has shown some activation of the KOR at concentrations of 100 nm, with an Emax of 22% at 30 µm. No plateau in maximal response EC50 was observed at concentrations up to 30 µm. Samidorphan similarly shows activation of the KOR in vitro, but to an even greater extent, with an EC50 of 3.3 nm and an Emax of 36%. As such, ALKS 5461 may possess both antagonistic and agonistic potential at the KOR. Because antagonism of the KOR seems to be responsible for the antidepressant effects of ALKS 5461, this property could in theory limit the effectiveness of ALKS 5461 in the treatment of depression. History. ALKS 5461 was granted Fast Track designation by the Food and Drug Administration (FDA) for treatment-resistant depression in October 2013. During June and July 2014, 3 Phase 3 clinical trials were initiated in the United States for treatment-resistant depression. Alkermes reported that the first 2 trials failed in 2016. In August 2017, based on the third trial, Alkermes announced the initiation of a rolling submission of a new drug application for ALKS 5461 to the FDA. On 31 January 2018, Alkermes submitted a new drug application for ALKS 5461 to the FDA for the adjunctive treatment of major depressive disorder. The submission was accepted by the FDA on 9 April 2018 after initially serving a refuse to file letter due to insufficient evidence of overall effectiveness. See also: Kappa opioid receptor section antagonists, List of investigational antidepressants. References. Further reading: Peshinya M., Karp J. F., Matthew S., Totenkopf M. S., Eric E. Zubieta J. K., June 2018. Endogenous Opioid System Dysregulation in Depression, Implications for New Therapeutic Approaches. Mol, Psychiatry. doi, 10.1038 per seconds 41380-018-0117-2. PMID 29955162. External links: Buprenorphine, Samidorphan, Addison site.